All right, Bang. Today's Thursday. It is April 24th. Welcome to the Dog Walk, presented by Barstool Sports. Today we are joined in studio by Rob Quinn. He is a biochemistry/microbiology professor at Michigan State. Rob, you joined us, I think it was back in December.
Mm-hmm.
We talked a lot about the gut biome and whatnot.
Indeed.
You are back. Thanks for coming back.
Thanks for having me, guys. I'm excited to be here.
Quick train trip.
Yeah, I was saying the train here leaves right from the Michigan State campus.
Piece of
So I
cake.
really get dropped off.
How many hours?
It's under 4, but then you change the time zone. So it's, I think it's like...
So I can leave day of for Michigan State night game and get there?
You could, totally. Get like a 3 o'clock kickoff. You could tailgate, watch the game, be back in time.
That's not bad.
Pretty good. Good to know.
How's everything going? Did you enjoy your first appearance? You're back for a second.
This was great. Yeah, I'll say I was a little nervous. Haven't done this kind of thing much before, but yeah.
Feeling good now.
Feeling good now.
Yeah.
Yeah, and everyone took well to it over there.
Yeah, it was great. I got a lot
Still
of
have a
good
job?
feedback.
Yeah.
Mostly my mom thought it was great. So.
That's the type of
good.
feedback I get.
That's
too.
really what you need. Yeah.
That's good. Well, yeah, that's kind of what we got into a lot last time. This time, I think we're going to do some DNA stuff, some stuff on cystic fibrosis, which I find particularly interesting. I have a couple friends who have had it.
Yeah.
It's a nasty, nasty disease.
Mm-hmm.
Yeah, well, why don't we start with what it is and
Sure.
what the symptoms look like, and because it used to be a death sentence, right?
For sure.
Early death sentence,
Right,
too.
yeah.
I was even reading like some of the really old literature in the Middle Ages. It was first described in like 1500s of a child with a sweaty brow. One of the symptoms of CF is you have a high salt concentration in your sweat. It's actually one of the diagnostic tests for a baby. Yeah. It has to do with what causes it. So it's a genetic disease caused by mutations in a gene, which combo genes as. It's all connected, right? Like some of the context of the DNA stuff is important to understand. But genes called CFTR, the cystic fibrosis transmembrane conductance regulator, which doesn't mean much to us, but so if you have a child, if you're a carrier, so your mom and your dad make a baby, you have 2 copies of each gene they get from their mom and the dad. So if you're a carrier of cystic fibrosis, you have 1 copy with the mutation in the gene, and the mom and the dad has 1 copy of the mutation of the gene. There's a 1 out of 4 chance the baby gets it because they swap their chromosomes, which we can still talk more about that stuff, what all this jargon means, and then there's a 1 out of 4 chance that the baby has CF, and a 50% chance the baby will also be a carrier. So some of the numbers are crazy. 1 in 30 people, particularly Northern European descent, actually Caucasians, white people, are carriers. So like if you're sitting in a classroom, maybe even in high school, and there's 30 people in the room, 1 of the people in that room is likely a carrier of CF. So if, the high school sweethearts meet each other in that room, or maybe in another class, the probability of them having a baby goes up. It's like 1 in 3,000 births in the United States has CF, which those numbers seem like still crazy to me.
Yeah, because I think I've known 2 people, not anyone close to me that had it, and it was in college. So is it something now that they will screen the parents for or screen the baby for?
Yeah,
Yeah.
definitely. I don't know all the details in like particular hospitals. I think it sort of depends on health insurance and certain hospital rules and stuff. But yeah, you could definitely test for it. And they'll do that for couples that are looking to have a baby. You're a carrier. Well, now there's a 1 out of 4 chance if you're having cystic fibrosis, which is, as you guys said, a terrible disease.
Is there anything that can be done if you know you're both carriers? Or?
Well, I mean, a whole other aspect to this that I was excited to talk about that maybe we should get into a little later is there's this new treatment. I won't use the C word. C word's a big word around these parts. Cure, because it's not. But man.
But like a symptom manager.
It's even like the greatest symptom manager.
Oh, good.
Symptom managers maybe ever. Yeah, it's incredible.
And how recent is that of?
2019, it was approved. November 2019.
Wow.
Yeah, the story of that is called Trikafta. Here we were talking about England. In England recently. It's called Kaftria for some reason in England. But yeah, it's a new therapy that treats CF. And I know people with CF, and it's like the comments I've heard is like, I've never felt this good ever when they take this drug within hours of taking it.
And just for people who don't know or aren't familiar with cystic fibrosis, so it's a disease in your lungs, right? It's not just a sweaty brow. Sweaty brow is not that big of a deal.
It's
so how does it
everywhere?
Yeah. So how does it manifest, and what makes it such a killer?
Yeah. So CF, again, there's a mutation in the CFTR gene. You have the CFTR gene in every single one of your cells. Okay, that's how genes work. We have the same DNA essentially in all of our cells, but which genes are being used in which cell sort of defines what that cell is. So CFTR is used in your lungs. It transports salt.
Okay.
Some complicated concepts that aren't great over a podcast, but essentially you're trying to balance the amount of water and salt in your cell and outside your cell. And CFTR does that. If you have a mutation, which is a change in the DNA sequence, it screws up the protein, the gene that becomes a protein that functions to do that. Now, CFTR is also expressed in other places. So the pancreas, the male vas deferens. So sadly, males with CF are sterile. Other places, the sweat, the skin.
Always sterile?
I think so.
Yeah.
Yep, I'm pretty sure. I wouldn't have to look it up myself to be 100%, but yeah, I'm pretty sure the women can have kids. And the Trikafta, the new drug, has really helped that. It's been like super awesome for reproduction for people with CF. But yeah, so the gene is present in all your cells, but only expressed in some. Therefore, the disease only presents in some. And one of the interesting facts that most people don't know about cystic fibrosis, even people familiar with it, is the name actually comes from the effects in the pancreas. So the cystic fibrosis in like the 30s, when they would have a baby, 2 years old, that would die, they had sweaty sweat. That was something that they knew, again, since like the Middle Ages. And his baby would die prematurely, a terrible story. And then they would do an autopsy and see this disrupted pancreas, these lesions, these fibrosis in the pancreas. And that's because there were no treatments at the time. I think one of the reasons CF is such an interesting subject to many people is it's such an example of the progress of medical science. So in 1930, if you were born with that mutation from your mom and your dad, you were unlucky enough to be one of those kids, you might live till 2 years old, maybe even less.
Oh, that's it, huh?
Yeah.
Okay.
Because the pancreas was so screwed up.
Yeah.
So you couldn't absorb fat. There's complicated reasons for that. And then you would die of malnutrition, essentially, of oily stools, things like that. But then they figured out, it's a lung disease, like you mentioned, but only because they were able to manage the dietary problems in infancy over the first 30 or 40 years of treating the disease. Then it became a lung disease later.
Oh, interesting.
Because it you couldn't even live long enough for the lung infections to be a problem.
Okay.
Yeah.
So 100 years.
Yeah, the
Now
30s.
where we are.
We're getting close to 100 years.
Essentially, man. And then as it's gone on, I think the 90s into early 2000s, it still felt like it was in the 20s, right? Life expectancy, maybe the 30s.
I remember hearing if you made it to 35 with CF, that's kind of a miracle.
Yeah.
It's amazing. I mean, I've been studying CF, let me think, so since 2012, San Diego. So 13 years going on. Yeah, something like that. And even when I started, I remember the life expectancy was maybe 40.
Okay.
And now it's yet to be determined, but this new drug came out in 2019, so it's been 5 years. But projections are that people with CF will live essentially full lives.
And what are the symptoms again, essentially?
In the lung.
The lung symptoms are gnarly. So people with CF when you're young, it's less of a problem. But again, these lung infections, and it's not really understood why, but they really develop in adolescence when you're in your early teens, and they get these infections. So everybody's had a lung infection. You get a virus or a bacteria and you're coughing up gross mucus.
Yeah.
Imagine coughing up more mucus than you ever have all the time, every day. That's what cystic fibrosis is. It's terrible amount of lung buildup, primarily from infection. But the infections happen because that gene isn't working right.
Yeah.
And it leads to this thick, sticky mucus that normally the infections, we're breathing in microbes all the time. Right? I'm spitting microbes all over this microphone, which is gross to think about, but true.
It's my microphone, so thanks.
Okay. Yeah. We're sharing germs.
It can't be worse than what happened to that microphone last week, so it's okay.
Okay. I don't
Guy
remember.
just sneezed right into
Right
it.
in.
so-
Really?
Yeah.
Pick that up on the sound for sure. Right?
Yeah, exactly.
But yeah, so because they don't clear the mucus out, they're very susceptible to infection, and the infection gets in and it stays there.
Okay.
And is that so? You're not going to use a C word like you said, but 2019, how did they find a way?
I mean, that's a hell of a story too. And it really speaks to the, what's great about pharmaceutical companies, medical science progress, but also some of the scary part, how long it takes. So I remember, I won't use the guy's name, just there's no real reason. We can look him up. But person when I was in San Diego that really started this research. So they were trying to find drugs, compounds, molecules essentially that would help fix this CFTR. But the way they did that, this is 30 years ago, maybe 25 years ago. So you take a little petri dish, you can grow human cells in a petri dish, and they're looking for the cilia. So your cells have little cilia that help clear that mucus out, and they beat like this. You can see it on a microscope. So people with CF, their cilia gets collapsed and they don't beat because the mucus is screwed up. They can't beat. So they would like take literally these drug libraries, they call them. So there's thousands of compounds. They don't even know what these things do. And they would put them over these cells to see if the cilia start beating again. So like 10,000 drugs, it takes years. They find a few. Like, holy shit, they're starting to beat again. Oh, these are interesting. This is like I said, like maybe 25, 30 years ago. They do those experiments and then they go into a mouse. And they say, okay, if the mouse has CF, which another complicated story, is not a very good mouse model for CF, but will it kill a mouse? Will this drug picked off the shelf kill a mouse? No. Okay, that's good. Let's go. Does it work? Does it help the mouse with whatever the disease is? Yes. Then you go to clinical trials. And this stuff takes decades, right? It kind of sucks because, man, it would be great if it would happen faster, but the safety is such an important thing that you're going to give thousands of people this molecule. What are the side effects? You don't necessarily know until you go through all these clinical trials. And we talked a bit about clinical trials and stuff last time. So then, stage 3 clinical trial, which is the final like safety and efficacy, like does it work thing in humans, large scale. That was done at probably like maybe 2015, I don't know the exact dates, but then the FDA goes through all the paperwork to approve it. And then bang, November 2019 was incredible. And there's a bit of a personal aspect to this, which is almost kind of funny. I started at Michigan State in 2018. And I sat down that fall and wrote what we call an R01 grant. So an R01 grant is like the big federal grant to the NIH. It's 12 pages, single spaced. It's like torturous. The whole thing is like 120 pages of, here's my science, give me money.
Okay.
And I sit down to do that in 2018. I submitted to the NIH and miraculously it got funded. So very early on when I started, I got over $2 million to study cystic fibrosis. And then get the money that summer. So summer 2019. And the word sputum, which is a word I haven't used yet, but that's the term for the gross mucus that gets coughed up, might have been in that grant like 5,000 times. And as soon as Trikafta came out, like 4 months later, they couldn't produce sputum anymore. The drug was so effective that this classic like mucus thing that they did was eliminated. So I've got a 5-year grant to the NIH, which just ended. So I guess I could talk about it now, with like millions of dollars to study this thing that all of a sudden like was kind of gone.
Wow.
And then COVID hits. So then no one can go to the clinics for like 2 years. Yeah. That was quite the experience, but that's.
Yeah. So just backtracking a little bit, when they first started messing with the cells in the petri dish and they're trying the library of drugs, and maybe you said this, but was that intentionally for CF? Because it's like sometimes I feel like, wasn't Viagra like they were trying to, they were studying it for something else and they just noticed that everyone got rock hard. Yeah. So but this was intentional, correct? Okay. All right.
I'm not as familiar with the Viagra literature, but yeah, definitely. So they had cells that have the same mutation. And that was the experiments were on these cells that have the CFTR mutation. Okay. And they don't beat. And you got like 10,000 experiments and like 5 of them made the little cilia start to beat again under the microscope. There you go. And then clinical trials, and a lot of them fail. They're not safe. Don't work in people. But 3 of them did. And Trikafta is called Trikafta because it's actually 3 drugs.
Okay. So then now that it's approved by the FDA, is this now like who owns that? Who owns like the patent and all that? Like who is administering this?
So the company is called Vertex. There was a lot of investment from the Cystic Fibrosis Foundation, which is an awesome foundation of basic research and promoting happy, healthy lives for people with CF. That's where a lot of the money came from. And Vertex owns the patent, as far as I know. I mean, I don't know a lot about it. I don't work for Vertex. Yeah.
But is it just like widely available to
that's anyone? Complicated. I don't know a lot about it, but it's expensive.
Okay.
Right. So like I think I heard initially it was like $300,000 a year.
Whoa.
Okay. Yeah. So your health insurance, and it's a battle, right? And it's, I think, one of the worst sort of injustices. I mean, that's not the right word, but is people who have CF and this drug is there, but they can't afford to take it. Like that's the saddest thing ever.
That's awful.
That is bad.
So what?
It makes the symptoms go away, you said, right? But
Yeah.
does it like...
There's still some, but wow, like the effect. And I've known people who have had it and I've talked to them, and just, if you're ever in a bad mood, if you're ever in a bad day, just Google Trikafta on like YouTube and just watch the videos of people talking about taking it. It's just like the most uplifting thing you've ever seen.
Really?
Like, they're living the most difficult life you can imagine, coughing up mucus every day, and all of a sudden they're like, oh my God, I feel incredible.
How many doses does it take for?
I don't really know that.
You don't know that, okay? But is it like, or not even a dose, but like a time frame? Like, do you have to take this medicine every day, or is
Yeah,
okay.
I think there's new formulations where it's once a day, but I don't really know how often people take it.
This is maybe outside of the 300,000 thing shocked me. How do they come up with pricing for these drugs?
I mean, you might have to bring in an economist to answer these questions better, but it took 25 years to develop.
Yeah.
It's a real challenge for pharmaceutical companies, especially for rare diseases. Even though CF, you guys know people with CF, it's still considered like a rare disease. Only 30,000 people in the United States that have it.
Okay.
So like for a company knowing that they're going to try to fix this disease, it's going to take them 25 years. They're going to have dozens and hundreds of employees in their company devoted to it. And then it's finally approved. And then, how do you make money off of that? I don't know a lot about it.
But what's the next step, though? Isn't it kind of like giving something to people with the gene? So, like, effectively, we don't have people who have it
gene
again?
therapy's coming. Yep. I suspect that eventually, I don't know when it'll happen. It's like some people predicted it would have happened by now. But CF is a major target for gene therapy. You just go in and just fix it.
And they have now totally mapped the entire genome, right? Like you can see, know where every single functionality.
You can certainly identify all the genes. Some of them, it's not totally known what they do yet. It's sort of studied independently. But the human genome is pretty.
And how many? Do you know how many there are? Because it's like how many?
Like 20,000 genes in the human genome.
Okay.
And this is all this like junk DNA.
What's junk DNA? I've never heard
that's
that
a
before.
crazy thing. So the human genome is 20,000 genes. It's maybe like, I think it's like 60, 70, 80% of it is just nonsensical. It doesn't seem to code for a gene. It's actually cut out when it's used. It's a big mystery in biology why so much of our genes don't seem to code for anything. It's weird.
It is weird. So is it something that's like, could it be like an evolutionary thing? Like, what are the theories about?
I talked to this in my biochemistry class. Shout out any listeners in the biochemistry class. It's not really known. It's like speculative why some things we figured out, like there's certain regions that we sort of thought were non-coding and didn't do anything. Oh, they have a function.
Okay.
But there's clearly regions, massive stretches of the DNA sequence in our genes and every what's called eukaryotes, which are everything but bacteria, essentially, that have just nonsensical, and it's literally cut out when it's used and just like
discarded.
Yeah.
Wow. Well, it's amazing that is here now. I mean, I know, like I think Boomer Sison's son has it, right?
Yeah.
Gunnar? Yeah, and I think Boomer's foundation was all towards CF.
Yeah.
So. Hey guys, let's pause this interview because I want to talk about my friends at Ghost. I'm in here right now. I'm drinking that sour strips flavor. I love this flavor. It's awesome. I think it's got a great can too. It's almost got like a funfetti white with like all assorted color droppings all over it. There's green, there's blue, there's red, there's yellow. It's awesome. And obviously it goes with the rainbow sour strip. So go check out that flavor, and you could check out the Straw Bango. It's here to stay permanently. It's now sold exclusively at Circle K. Ghost, listen to you guys. Everybody wanted this flavor back, so it is back. It's a perfect duo of fresh strawberry paired with sweet and sour mango for a crisp and refreshing flavor for any occasion. It's like other Ghost Energy. It's always fully transparent, fully loaded, feel-good energy. 0 sugars, no artificial flavors, 200 milligrams of natural caffeine. Now sold nationwide at Circle K stores, and it's here to stay. Go check that out. Straw Bango. Shout out to Straw Bango. I got to go grab a Straw Bango right now, actually. So shout out to Ghost. Go have one.
I thought if we can, I'd take a moment to tell a story about my friend Mike.
Let's hear it.
He had CF, and he died. So I moved to San Diego to do a postdoc, which I might have talked to you guys about last time. Postdoc is like the time after you get your PhD, then you get more research training before you apply to be a professor. And I did a postdoc in San Diego for like 6 years. And the lab that I joined, this is really quite the story. So the lab that I joined is sort of reflects my existence now, where we talked about gut stuff. And we also work on coral reefs in my lab and cystic fibrosis, which like makes no sense. And it's kind of juggling.
Does not make sense.
No, it doesn't.
Yeah.
My PhD was on lobsters and that makes less sense.
Eddie,
I get it.
Eddie sent me the screenshot of the topics. I'm like, okay. Coral reefs and DNA. Let's go.
It's pretty unique because it does reflect the lab I came from, but there's a very particular reason for that, as far as I understand it. So I joined the lab to study cystic fibrosis in 2012, I guess it was. Maybe even earlier than that. And when I got there was this guy, Mike. He's from Texas. I won't use his last name. It's not important. But he sadly died while I was there. So Mike had cystic fibrosis, and he joined the lab. And the whole reason that this lab, that really was a coral reef lab that studied microbiology of coral reefs, particularly something called a bacteriophage. You guys ever heard of phage?
No.
Never heard of them? Some people have heard of that. Bit of a hard concept, but well, not really. They're viruses that infect bacteria.
Okay.
And there's literally multi-trillions and trillions and trillions of them on Earth. So viruses infect bacteria. So they were studying like bacteria and phage on coral reefs. So Mike finds out about this lab. He's lived in San Diego. I think he was from Texas originally. That was very clear. He was very Texan. You know what I mean? So Mike finds out that there's this phage lab, this bacterial virus lab at San Diego State University. And he's like, I want to go there to study phage, because he believed that phage were going to help his cystic fibrosis.
Okay.
Because he was going to take the phage into his lung and kill the bacteria, which is absolutely a thing that people are doing, including in San Diego. People that I collaborate with are doing this exact thing now. They're treating people with CF with these bacterial viruses to kill the bacteria in their lung, and the viruses don't harm us. They can't infect our cells.
Oh, interesting.
Okay. So the kind of long story about Mike, I mean, he was an amazing guy. He was an absolute badass. And he would we'd sit in this big office, like maybe 3 times the size of this room. There'd be like 20 people in there. And Mike I have to get samples from him. So I was a cystic fibrosis guy, and half the people in the room are studying coral reefs. So I'd ask Mike for a sample, and he would cough up his sputum into a tube, like right in the middle of the day. And people were just sitting next on their computer. And it was one of the most horrific things you ever heard, the coughing he would do.
Yeah.
And it was just typical for him. That's how he lived his life, which just kind of speaks to his badassery. And I did that for a long time. And then ironically enough, and we're going to this meeting again in a couple months. So we were in Colorado. I mean the people I worked with there, our collaborators, some of the medical doctors we worked with, were in Colorado at a scientific meeting to talk and research and discuss what we do to study cystic fibrosis. And we get an emergency email to his physician. So Mike's physician was with us in Colorado, that Mike's in the ICU. And he was really doing badly. He'd had a few events before that were serious. And we're like, holy shit, oh no, Mike, we're not there. At the time, we had set up this program we called Rapid Response. This omics data stuff I talked to you guys about last time, DNA sequencing and all that stuff, we could do it pretty fast now. The turnaround times are a day if you really force it. So it could be relevant in the clinic. So we're thinking the part of the plan was that when these events happen with people with CF, we would generate all this data as quickly as we could, try to interpret it and help the physician treat. And we tried to do that with Mike, but we weren't there. We were in Colorado. Meanwhile, it didn't matter. So we still tried to do it. So on our computers, getting samples from Mike in the hospital. He's got blood in his lungs. He's doing terribly. And we get all this data, and we're trying hard to interpret it in real time while he's essentially on his deathbed. And then he passes away. And we didn't make sense of the data till later. It actually, the most tragically ironic thing, and the truth is we'll never know, because when you do things that quickly, it's very difficult to control for variables and do it in a controlled way. But the data was showing us that he had what we think was an E. coli infection in his airway we'd never seen before. We don't really know, but that's what the data had said. That E. coli had something called the Shiga toxin. You ever heard of the Shiga toxin?
No.
That was dysentery.
Okay.
The Shiga toxin is just another gene the bacteria has that is bad news in the E. coli. And that Shiga toxin is only there because a bacteriophage, that same virus, infected that bacterium and brought it with it.
Huh.
So the tragic irony that, again, is speculative, we may never know, but that Mike came to that lab to study phage, hoping that it would help him. And it may have been a phage that killed him.
Wow.
Huh.
Pretty crazy.
Yeah, that is.
But you said that people are trying to use phage for this exact purpose now.
Yeah, literally as
okay.
Yeah.
So would that be, so you have this drug concoction that works, and then are they doing the phage hoping that that'll be a cheaper option, or what is
it is cheap? Yeah. Phage will just reproduce themselves, so you grow phage on bacteria.
Okay.
You purify them and spray them into someone's airways. It's been used systemically, so like through your blood. There's incredible stories. This is also at San Diego. This is like maybe 8 years ago now, where a person, I think he was a medical doctor himself, went to Africa and got like a horrific infection. It was Acinetobacter. It's not really important, but it's like terribly antibiotic resistant bacterial infection.
Okay.
He was on his deathbed. And they like had this massive effort similar to what I just told you. I wasn't involved in this, but some people in my lab were when I was there. And they like scrambled to find phage that will kill this bacteria in this guy's lung, purify them, give them to the patient, and he survived.
Wow.
Yeah, it was like deathbed. This incredible. There's a book written about it actually.
So can phage work for any sort of bacterial infection? Is it like
theoretically? Yeah. There's all kinds of complicated stuff about specificity. So you can't just take any phage and infect any.
It has to be. Yeah.
There's like this interesting dynamic between the bacteria fighting against the phage fighting against the bacteria. That's also where CRISPR comes from.
Really?
Yeah, it's a whole other story.
So well, we got time.
Okay.
Yeah. So what do you mean that's where CRISPR comes from?
So the enzymes and it's pretty hard to explain actually, but bacteria are fighting against these phage all the time, much like we're fighting against viruses in our bodies. And they have these defense systems where there's like enzymes that will cut DNA that they know is phage. And they do that with little other pieces of DNA that will like target it to a particular sequence.
Okay.
Which 20 years later, Nobel Prize, or more than that now, because we've been able to harness that targeting mechanism and that cutting mechanism to cut and edit. So like DNA editing and stuff like that's really just like arts and crafts with DNA. You like have a little sequence, you target what you want to target, cut it out, fix it, and that's what CRISPR is in a very basic sense.
What's a horrible disease that is being worked on tirelessly right now?
I think sickle cell anemia is getting close to being maybe, again, the C word is dangerous, cured from things like CRISPR and gene editing.
Yeah.
Cystic fibrosis could be fixed by that too. Yeah.
What does ALS fall under?
Don't know a lot about it. That's the myelin. It's like a neurodegenerative disease, also genetic. Don't know a lot about it.
Yeah.
But
okay.
Yeah, that would be one, man. If they could cure, that's one of
the worst. Yeah. That's why
the worst of the worst.
Yeah.
That's such a terrible thing.
And with like CRISPR, am I confused that that's like the people say like, I'm gonna have a designer baby, like we're just gonna pick out we want this baby to have blue eyes or whatever feature.
I mean, theoretically,
these
things are possible. Who knows if we're doing the podcast 50 years from now with great
You think it would take that long?
Oh, I don't know. God, things move fast.
Yeah.
It's so hard to predict that.
Do you guys in studying DNA, do you guys have like AI software that helps you comb through everything?
Absolutely.
Yeah.
Yep.
And
probably in my lab right now, people are doing that.
So when did that become more prevalent, AI with studying DNA?
Yeah, it's longer ago than you think. Like AI, artificial intelligence, that language model stuff is relatively new. People have been working on it forever.
Yeah.
Like some of the most cited, I was actually reading a thing in Nature just yesterday, but the most cited papers of all time. Some of them are some of the early language models that were developed in like the late 90s.
Okay.
But to answer your question, so that basic idea of pattern recognition in data using machines, I was doing in San Diego. People have been doing for maybe 10, 15 years before that.
Okay.
The language model thing, predictive text, that's relatively new.
Okay.
And
but combing through data is not anything.
Yeah,
that's just like software. That's just a computer. But that's considered AI too.
You say machine learning. Machine learning and AI, I guess, are synonymous in many ways. But we use machine learning because those thousands and thousands and millions and billions of sequences and base pairs. I can't look at that with my eye. It's like reading Tolstoy 10 times and trying to find a particular thing in it.
Yeah, a needle and a stack of needles kind of thing.
Exactly.
That is interesting though. And is there is there more work being done? Do you think like in 10 years, like we'll have AI be like, yeah, like this is the problem with this gene and this is how to fix it?
I think, yeah.
Yeah.
Sure. That could be an example. I'm trying to think, like, I'm thinking out loud a little bit, but
That's all we do here. So feel free to do
it's
that.
spontaneous.
Yeah.
Sure. It's a bit scary to a scientist.
Yeah.
I think things like that where data mining, that's probably where the first big steps will happen. Like we talked about last time, the gut stuff. Right? What are we supposed to eat? I told you guys eat your prunes. You've been eating your prunes?
I haven't had a prune.
I gotta get on that.
Yeah.
Remember to eat your prunes.
Okay.
Chia seeds. I actually started, you mentioned those. I started eating them since then, last time we were talking. So like we could mine clinical data. You can do that. So, there's all this HIPAA stuff, de-identified, whose data it
Right.
is, and there's ways to do that. So you have like clinical records from decades of treatments. How do you find associations with people that had diabetes and XYZ, or like, so machine learning is really good at stuff like that, where they can say, oh, if you have this life history, this medical history, this genetics, and that other thing, you're really likely to have this. So avoid that. That stuff's coming if it isn't here already.
Okay. You want to get into coral?
I kind of want to get into the direwolf stuff first.
Okay.
Oh God.
So
I listened to that episode.
You saw that? So like obviously DNA, you're a DNA guy. What are your thoughts on that? Would you say it's an actual direwolf? Because I know that's some of the pushback that like Ben and his group are having.
People are mad about that. Yeah. They're mad about the terminology.
Yeah, I think some scientists are mad because is it truly a direwolf? They edited the DNA. I honestly don't know a lot about it. I've just like read the articles you guys probably have.
Yeah.
But if you just kind of tweak the sequences, the DNA, I think we could talk a bit about what that really means too. It's not necessarily really a direwolf, but I guess it would be like direwolf adjacent.
Yeah.
You're becoming more like one. But it is incredible what this company is able to do, and there's a lot of basic science people doing it too, where you can like mine this DNA that's been on Earth for thousands of years.
Yeah.
There's an incredible story of a lab, I think it's from Europe, where they got DNA out of soil from like Greenland, like dug through ice cores in Greenland and
like
pulled out
permafrost.
soil.
Yeah.
Permafrost soil. And they sequence everything. And you just see like woolly mammoth DNA and like all these crazy organisms from like, I think it was like 1 million years ago.
Jesus.
It's like a look back in time. It's incredible.
So it's that makes me think that like we will get dinosaurs back. So we will have Jurassic Park.
So you know what's funny? Like Dolly the sheep. Right?
Yeah, the cloned
I think
sheep.
I was in undergrad. I don't know what year that was.
I was little. I remember that, and I want to say it was like early 90s.
Yeah.
So
I don't remember that.
So I think it was in the UK, right? And they cloned. They cloned. It was like the first ever thing that they cloned. They cloned a sheep.
And I don't have a clone. Do you guys?
No.
That's what I know of.
I could use one.
I wouldn't want one. There doesn't need to be more than one of me.
If it was subordinate, it'd be good.
That Black Mirror episode scared me enough, so I'm good.
So what's your point of that though? Just because we can do it?
I guess what I was gonna say is like there's examples where the Dolly the sheep. So we had cloned sheep 30 years ago. Mm-hmm. Maybe not that much.
No, I think that's probably about right. Yeah.
And it's not like a thing where we had text messaging 30 years ago, and now it's like everything we do all the time. So I don't know. Some science progresses really quickly, and others doesn't. I don't necessarily know why.
I wonder if it's like there's not enough of a benefit of it. Like just make a sheep the old-fashioned way. Get two sheep together.
Yeah, that's the one way you can make a sheep.
Yeah.
Put them together at the right time of year, and you get a baby sheep. Yeah, that could be it.
Yeah.
But I don't really know if much has followed from that. There was a couple stories that I don't know a lot of detail about, but there was some scientific misconduct that went on since then. Famous example, I think it was in South Korea, where people were doing this cloning stuff and just made up a bunch of data and didn't really publish a paper, and it was like not real. Maybe that slowed it down a bit.
How often does that happen?
Not very often.
No.
Credit to us, I would like to say, for not doing that, because there are thousands of papers published, thousands of journals all over the world, and the retractions and the true misconduct, like intentional misconduct, is extremely rare. Like you hear one a year maybe.
Yeah,
one a year.
Yeah,
and
mistakes happen.
Sure, mistakes happen, but you say one a year.
That's kind of guessing, but
sure. But then you read about like, so then I guess who is doing that and to what end would be like, what's it like that the people in South Korea, like what are they?
It's a lot of pressure.
Yeah.
I mean, it's a pretty crazy thing. Like when I got my job at Michigan State, you have this tenure clock, this timeline to like produce, be awesome, publish, have a great lab, diverse lab with people doing awesome science and get grants. A lot of pressure. And you could see how people would just sort of shortcut that.
Okay.
And that's happened. There was a pretty famous example within the last couple of years in, I think it was physics, Rochester University. Real true misconduct. Like misconduct's not really the right word. I'm kind of blanking on the
fraud.
Yeah.
Yeah.
Just basically made up data.
Okay.
Wasn't reproducible. And that's where you get caught because people can't like do what you did.
Yeah.
They're like, how did you do this? And you're like,
But shouldn't everybody know that?
What do you mean?
Like, if like you're fudging the data, you're committing fraud, and you know that if you publish this paper and you get the initial pat on the back for having this breakthrough on whatever you're studying, you know that people are going to try to replicate that. So it's like your timeline to be like, it's like the benefit doesn't outweigh.
Yeah, that would be truth to that. I mean, there's that deterrence, right?
Yeah.
The Theranos girl, what was her end?
Yeah, Theranos.
What was her
well,
end
she
game?
was
Making money.
being a billionaire.
Yeah, for sure. But like at some point,
Well, there's a lot of every university.
But that's different than academia.
Yeah, for sure.
Yeah.
I'm just as an example.
Every university in the country, including mine, has extensive training at very early stages of people getting into research. I had a responsible conduct of research. Like you need to report your data. You can't fabricate things. It's really well organized and communicated well early on. Hopefully that helps.
Yeah. How do you guard against, I guess, when you're going through these studies, like your own confirmation bias? Like you're looking for something and it's like, I feel like that's,
hard thing.
Yeah, where it's like, I want it to do this, and then you look for ways to make it.
It's a hard thing. It's happening right now in our coral work. We have this new hypothesis about this enzyme we think is important, and then all of a sudden it's like not really matching what we thought, and you just have to be
willing to throw it away. Yeah, because I can imagine that, like, you develop this hypothesis, that takes time.
Yep.
Right.
Sure.
All the reading, all that, and then you go and actually do the study, which takes time and funding and everything, and then it's like you're so far down the road. How long, if you're doing a study on a particular hypothesis, what is the time frame from like, all right, we have this theory to the end? Like
wildly varies.
Okay.
Yeah. We had a paper when I was a postdoc in Nature that was simply like, there's a lot of luck involved, right? Like we did this pretty complicated experiment, huge data set we were looking through with these mice, and all of a sudden I like found this thing. My boss was with me at the time, and like, oh wow, look at that thing. And then 3 months later we submitted to best journal in the world. 9 months later it got accepted, and then we have other projects in my lab that we started 3 years ago and we're still slogging through.
Okay.
It's very variable.
Just depends.
Yeah.
And listen, like you said, once a year for like, those are like straight up fudge numbers on studies. What about you hear about some of these studies that are maybe funded by a certain place that is looking for a potential result?
Oh, that's a thing too. Yeah. Conflict of interest. I mean, I have some conflicts that develop at times where, like, if I consult for a company, I have to be quiet. I can't talk about their stuff. I mentioned that last time. If I start a company, which I might do, then there's a real conflict, right? Where it's like, oh, well, the work that I did is awesome. Would you like to pay me to do it?
Yeah.
That stuff's really, you have to be very careful.
Yeah.
It's like, right, you hear that a lot where it's like, oh, this study was funded by so-and-so company because they...
Right. They want to... It's like there was that famous, or it was like I think it was in the 50s. It was like the building of like the food pyramid. And you had a guy from Harvard who was out there being like, a calorie is a calorie. So if you have a calorie from,
an apple
and corn syrup, has the same effect on your body. And so he had studies that showed that, and then it's like that became the foundation for the American diet, which has had pretty damning effects. So, and then it's like now we're 70 years later, and it's like it still has done damage. It's like we're still kind of having issues with that as a country, because it's like we always hear that's like we spend the most on healthcare in this country, and have the worst results. Like something's not right there.
Yeah.
So it's like I don't know what the actual problem is or what the root cause is, but those two things should not be the case. If you're spending the most, you should have better health outcomes.
Yeah, if you think DNA sequencing is complicated, deciphering all that stuff is more complicated.
For
I'm
sure.
from Canada, where we have a free healthcare, very different system.
Yeah.
So I'm like, don't understand. Still, after living in the States for almost 20 years,
Yeah.
I barely understand how this all works.
Yeah.
I guess like how if that's once a year, how is there even an amount you can guess that conflict of interest happens?
Well, you hear that with pharmaceutical
Academia.
companies all the time.
I don't know. That's a good question. I mean, also there's big degrees, right?
Yeah.
Or like little harms compared to big harms,
Yeah.
where you're literally like fabricating things so that people invest in your company. That's bad news.
Yeah.
I don't know. I would be guessing. But the true scientific misconduct, like in an academic sense, is very rare. But there are mistakes. Mistakes happen. You can do corrections. Like, oh, whoops, we didn't
Fuck that up.
wrong figure there. Like, oops, that's actually not. Can we just correct this?
Yeah.
You got to do it.
Yeah.
Because it's an important thing.
For sure. We can get a coral reef.
Well, one last question on that. When you do correct it, if you, like you say, you make an honest mistake and you do it, and you're under all this pressure and you're like, I got to correct that. Is it still like a black mark on your record if you got something wrong and correct it? Or is it like, hey, like you eventually got it right. Thanks for correcting it.
My job can be pretty high pressure at times.
Yeah.
Those are the kind of reasons why. Especially because there are people that work for me. Shout out all of my awesome trainees and students and technicians. And I don't always know what they do. And it can be hard. You can't just like micromanage and helicopter everything.
Yeah.
And they may not know at times mistakes. It could be pretty pressure in that sense to not do that.
Yeah.
Even unwillingly. Yeah.
And one other thing on the academia front that you said last time, that it's hard to get people into science.
Yeah.
It's becoming harder to get people into science.
That's kind of why I'm showing up here.
Okay. All right. Yeah. You're the face of science for
Well, I kind of speak to someone who would listen. Yeah.
Yeah. Well, it's like we like that stuff, and I feel like Rogan has scientists on all the time, different types of scientists. So I don't know. Has there been any? It's only been 3-4 months, but have there been any steps taken at Michigan State to like encourage more people, or is there like a marketing program other than you coming on the podcast to be like, hey, like this is the benefit?
Not
this
really.
No. What would you do? What would you do?
I would do this.
You do these, yeah.
Yeah. Yes, and like anyone that'll listen. I mean, I was sort of motivated to like talk about my job and like what being a professor is. All of us, you guys went to college, and you told me, and like you sat on the other side of that, and just listen to this boring person blab on for an hour, and then do it again, and then tell you you're bad at it because you had poor grade. And there's a lot more to it. I advertise the position in a way like I absolutely love my job, like being able to like research and discover and have those moments I talked about in that paper we published, where like you see something is like a big deal. Holy shit!
What was that thing? Did you ever?
It's kind of it's a bit complicated. We discovered new bile acids made by the gut microbiome.
Okay.
That's part how I got involved in all this. Long story, like the first bile acid chemical structure was described in 1849. We just found in 2020 that a whole bunch of bacteria make like dozens to hundreds to thousands more. That was in Nature, which is like a big deal for me.
Wow.
Yeah. Wow. Congratulations.
Thank you.
Yeah.
That's very cool.
Yeah.
Lots of people helped.
You're teaching what again? Like,
I primarily teach biochemistry, like DNA biochemistry. I just finished actually.
To undergrads or graduates? Or,
Undergrads. I teach grad classes too.
Yeah. How locked in are the kids these days on all this kind of stuff?
Pretty locked in.
Yeah.
Yeah.
That's
It's
good.
like a gradient that you can visually see. Like the kids that sit in the front row are like listening to my every word, and then the kids in the back that are on their iPhone. So there's like literally a physical gradient, I think.
Yeah. Do you ever get like, is there ever kids in the back who are like, if you just try, you'd be amazing at this?
I don't know. I'm wildly impressed by the undergrads at Michigan State and really everywhere I've been. These kids are great. They're awesome. They're all capable. Nothing but good things to say.
Okay. Do you want to do coral?
Yeah, let's do coral.
Why are you studying coral?
Now, if we're going to be truthful with myself. So I got involved in corals when I was in that San Diego lab. It's not that interesting of a story, but basically I learned this technique that no one else in the lab knew how to do. I was studying cystic fibrosis, and they're like, hey, can you like do that thing on my corals? I was like, sure.
Okay.
And it led to a couple of papers. This is like 10 years ago, maybe a little more. And then when I started at Michigan State, you get this thing called startup funds, which is fun. They give you money to start your lab. And because I got that big grant from the NIH, I had all kinds of money. So I had a lot of flexibility. Usually your startup funds are like devoted to like specifically what you were hired to do.
Okay.
I had over $2 million from the government to do those things. So I had some money to do more fun stuff. And I contacted some friends that were in Hawaii. They were studying corals because they were in my old lab, and they, that's the cool thing about academia, which I imagine other professions is the same, where you have the people you kind of grew up with in your workspace. They go off to do their thing. Maybe different if you work for a company, because like companies don't really interact much, right? But in academia, you're like, hey, you want to do this thing together now that I have all this stuff and you have all that stuff? And that was in Hawaii, and I'm doing that ever since. Then we got a NSF grant 2 years ago to study coral bleaching.
Okay.
But also, like the truthful answer is I'm fascinated by corals. I have been, my PhD was in marine stuff, with the lobsters. I've been involved with that for a long time. But Michigan's cold.
Yeah, it's a tough place to study coral.
The Caribbean is not cold. It is Hawaii. So there's a lot of benefit there. We actually have corals in my lab now.
Yeah?
Growing in it.
So I feel like this was, it was like a big story, probably more so when I was a kid, like the Great Barrier Reef and all the corals are dying all over the place. So is that what you're trying to solve for? Yeah.
Well, I mean, solve is whatever we could do.
Yeah.
Yep, specifically.
And it's a living organism, right?
Yeah,
Or
corals are cool. They are a true symbiont. If you guys know symbiont, the term means.
We should just
Nope.
Yeah, sure. So it's like
assume we don't.
Yeah, sure.
And assume the audience doesn't either.
Okay, yeah.
I obviously know what it is.
Symbiont is when two organisms can really only exist together. They live together in this harmonious state.
Symbiotic relationship.
Yeah, symbiotic relationship.
Yep.
They depend on each other. Corals are very much like that. Really, all organisms are. We all, our gut microbiome, you can argue, are symbionts in us, right? They give us the good stuff when we eat our prunes. Eat your prunes, guys. What was I going to say? So the coral is a true symbiont. They have these algae that live inside their cells, and the algae photosynthesize and give the corals food, essentially sugar from that photosynthesis. And the corals host the algae inside their cells, kind of give them a harbor. It's a little unclear how the fair deal works out, because what does the algae get? They think it's sort of a happy, comfy place to live inside the coral.
Okay. And then the coral is important to the ecosystem. Why?
I mean, it is the ecosystem, you could argue.
Okay.
Tropical reefs, so they build the reef. So like a reef is, you've been to a tropical reef here, you guys?
I have not.
I have. Caribbean, yeah.
The Caribbean?
Yep.
So they're like rocky.
Yeah.
And they're just like little mounds of.
They don't look like they're alive, really.
Well,
when you're going,
you might have been to the crappy ones.
Okay.
The nice ones look pretty cool, like alive.
Well, right. Like
I know what you mean.
Yeah, they look like a weird kind of
and a lot
bony
of those rocks, and like what you see that is the reef used to be coral, and they have built it from growing. Because they deposit calcium carbonate. Like the joke we make in the lab is corals are just snotty rocks. You take a sample, it's like a rock.
Yeah.
Calcium carbonate, but covered in an organism.
Okay.
And they got mucus, much like that's sort of how the CF connection came in.
Wow.
Okay.
And are we doing better at preserving the Great Barrier Reef?
Yeah, I would say so. I mean, it's a big job.
Why was? Did they figure out why it was dying?
Well, it's dying because of climate change.
Climate change,
increased sea surface temperature, among other things. But there's a real concern about the rising sea surface temperatures in reefs, and the threat. Corals live in this pretty tight window of temperature, and if it gets too hot, they bleach.
Okay.
And the bleaching is they lose their symbiont. So that little algae that lives inside their cells, then this big happy kumbaya symbiosis, if it gets too hot, it falls apart. The coral can survive that. It can like kind of stick it out, but if it's too hard or too long and it doesn't acquire the symbionts again, they die.
Okay.
So these bleaching events, and there was one, I'm terrible with time. Was it last summer or the summer before? Yeah, 2 summers ago in Florida. Like some of the bleaching there, they had like record high water temperatures in the Caribbean Sea. It was like nightmare. These corals all bleached. All these people that I know that work there, like all their experiments are dead. Damn. Not all, but yeah, it's been really bad.
How have they combated that, though? You said
there's really cool work being done, including people we work with in Hawaii, in restoration. So you can like selectively breed corals or choose the algae that are like really thermally tolerant.
Okay, that's what I was going to ask.
Some of the stuff we're doing in our lab.
Yeah.
And then like you select the ones that are tougher, put them back on the reef.
And how prevalent are those just in nature if you're not selecting for them?
We're trying to figure that out.
Yeah.
Literally on the train up here, I was looking at data about
Yeah.
that exact thing, and we'd never seen it before. So I work in Curacao, in the Southern Caribbean,
Yep.
a little bit, and we're seeing
Little League baseball powerhouse, Curacao.
Yeah, who's do we know?
It's like Xander Bogaerts.
Xander Bogaerts and this Kenley Jansen.
Yes.
So last time I was there, I met Kenley Jansen.
Okay. All right. Yeah.
I told him to sign with the Blue Jays.
Did he?
My favorite team.
Yeah.
Did not.
No.
We got Vlad.
Yeah.
We signed Vlad.
Yeah.
Psyched
nice.
about that. I of course lost my train of thought with the
Yeah. You're down in Curacao. You're studying it on the train.
Yeah. So there's some of these symbionts they think are more thermally tolerant, tougher in high temperature. And I just looked at the data on the way over here, and the Curacao corals have some of these. And I'm not sure how known that was before.
Okay.
I looked at my laptop.
And then in theory, so you have these down in Curacao where it's more warmer than it would be in theory, at least my brain could be wrong, than it is around Florida.
Yeah, Curacao is pretty close to the equator.
Right.
It's hot.
Right. So, and if you just take a sample and transplant it, they're fine.
It's exactly the experiments that are being done.
Okay.
Yeah. And literally as we speak, we have samples from our team in Hawaii that there's a couple of different bays in Oahu. One of them is warmer than the other. And they can swap the corals to see how like the corals that grow in the colder bay will do in the warmer one. This is exactly like the basic kind of science stuff.
Okay.
Not basic in its design, but wildly complicated and effort to do practically.
And then for this type of experiment where you're going to Curacao and you're swapping it and you're bringing it other places, what type of funding do you need to run a lab like that?
Yeah. Money from the government.
Right. But like a dollar amount, like how yeah.
I don't know how much I really want to talk about the money numbers, but you get an NSF grant. I mean, it's fair to say it's probably common knowledge for everybody. You know, a good National Science Foundation grant are usually 3 years, $1 to $2 million, which is a lot, but it pays salary. Right?
Yeah.
So if you have like 3 people that work on the project, they make a reasonable salary. That money gets pretty quick.
Yeah.
So really, the grants pay the people to do the work. And then the cost of the experiments, which is sometimes minimal compared to the cost of the people.
Okay.
Well, that's good. It's in a better spot now than it was 10 years ago.
In the Great Barrier Reef, there's some people that are doing amazing work where they're selectively breeding these corals, but it's a big job, right? Like, talk about a drop in a bucket.
The reef's big.
But like a bunch of corals out there, I think at the size of the reef.
Have you been there?
I have when I was a kid. I was like 17.
And then, so you said you've been obsessed with corals for forever. Like, what was the thing that inspired that?
Well, I wouldn't say I was forever. It really was working in that lab. I was always involved in marine stuff.
Yeah.
Interested in marine microbiology, but working in that lab, it was just so cool.
Yeah.
The stuff they would show. And I'm like going to the hospital, like sputum samples and they carry them back. They're like, I was in the Southern Line Islands in the middle of the Pacific Ocean. Here's palm trees that I took a picture of. And I'm like, I was in the hospital this afternoon. So I was maybe interested in that.
Yeah.
And just explain why it's so important, the Great Barrier Reef and just coral in general. Like,
well, I mean, if you care about diversity and beautiful places and organisms and biological diversity, there's all kinds of economic implications. So particularly for reefs, which are often in countries that are less wealthy, like a lot of their people depend on fisheries.
Yeah.
And like healthy reefs have lots of fish. It would, it's an economic thing, but also just the idea of biology, like the dire wolf, right? Like, do we want to have all the corals just be like the dire wolf? We have to edit it back to make it exist again. Losing beauty of biology and organisms is a tragedy that we should avoid.
Yeah.
Yeah.
For sure. All right, then. I think I'm good, Rob, unless if Chief, you got anything.
No, I think I'm all set.
Yeah. Unless
Do you have anything,
else
Rob?
for us?
Nope, I don't think so. This is fun.
Yeah.
Pretty much all of it.
Yeah. Good.
No, that's all I got.
Sweet. Awesome. Well, thanks again for coming in. It was fun to chop it up with you. Is there a way people can find your
Maybe
stuff?
my Twitter at @Quinn_labs. Yeah, but they can Google me, Michigan State. People want to reach out. I have a website too. They can just find online.
Beautiful. So get in touch with Rob if you have any questions.
Quinn Labs.
maybe
Quinn_labs.
Yep.
I'm going to follow Quinn_labs.
Awesome. That's it, everybody. Thanks again, Rob.
Okay.
Appreciate you for watching and listening. So we will end here and we will see you guys on Tuesday.